Howdy guys, my name is Sofia Guerra. Today I'll be covering with my partner Mayra Landeros Lab Video Report 10 over Biochemical Test 4. Introduction. The purpose of this lab was to perform a biochemical test, urea hydrolysis, CAMP, and coagulase, to identify bacterial metabolic properties and potential pathogenicity. These tests are significant in distinguishing between gram-positive cocci and gram-negative bacilli based on enzyme production. Uricase activity helps detect organisms that can hydrolyze urea to produce ammonia, altering pH levels. The CAMP test identifies Streptococcus agalactiae based on hemolytic interaction with Staphylococcus aureus, while the coagulase test detects Staphylococcus aureus, a major pathogenic species, by its ability to clot plasma. For materials, the media we used were 4 urea tubes, blood agar plates, one agar plate, my bad, and a rabbit plasma tube, two of them. For cultures, we had E. coli, P. vulgaris, S. aureus, S. epidermis, S. agalactae, S. pyogens, and our two unknowns, which are 122 and 285. As far as our procedure, the urea test, each urea agar tube was streaked with E. coli, P. vulgaris, and the two unknowns, 122 and 285. Tubes were incubated at 37 Celsius for 48 hours. A color change to pink indicated a positive result due to increase in pH from ammonia production. The pH level of urea agar before a positive result is near neutral, around 6.8, but after a positive reaction, it becomes more basic, around 8.1 to 8.4, turning pink due to phenol red indicator. For our CAMP test, we followed an on-blood agar plate. S. areas was streaked in the center. S. pyogens was streaked above and S. agalactae below, without letting the streaks touch. The test was incubated at 37 Celsius for 48 hours. The streaking method allows observation of synergistic hemolysis. S. agalactae produces CAMP factor that enhances the beta hemolysis of SRAs, forming an arrow pattern at the junction, confirming a positive result. For our coagulase test, organisms were inoculated into 0.5 milliliters of rabbit plasma and incubated at 37 Celsius for 48 hours. Coagulase converts fibrogen to fibrin, forming clots that protect bacteria from phagocytosis, directly linking to pathogenicity by helping SRAs evade host defense.
Now we go into our results, starting with the urea test. So as you can see here for our E. coli, there was no color change, um, indicating that it was negative. For our P. vulgaris, there is also no color change, indicating that it was negative. Now for our unknown number 285, there was also no color change, indicating it was negative. The one that had the major difference was our unknown number 122, which it completely turned the whole tube pink, indicating that it was positive, that urease was present. Now for our CAMP test, all cultures showed visible growth, but as you can see in the middle, there was a distinct glow, which indicates enhanced hemolysis, noted on the SR streak area, indicates synergistic hemolysis and positive CAMP factor. Now for our coagulase test, the SRs is more noticeable to be more cloudy or clotted, indicating plasma formation, which also indicates positive that coagulase is present. As for our S. epidermidis, there was a slight cloud formation, so it could either indicate that it was weak or it was all in all just negative for that test. In conclusion, in this lab, unknown number 122 tested positive for urease production, while E. coli, P. vulgaris, and unknown number 285 were negative. The CAMP test demonstrated characteristic hemolytic synergy with SRs, confirming the presence of CAMP factor and S. agalactae. The coagulase test verified that SRs produces coagulase, contributing to its pathogenicity by forming protective fibrin clots. These biochemical tests collectively help identify bacterial species based on metabolic properties and virulence factors. Thank you.